Hello, my name is Michaela Sanders, and I will be discussing the protein type 1 collagen and its mutations. So, a normal function of the collagen protein is to provide support for organs and to strengthen tendons and tissues. And all collagen is a structural protein because it provides support and structure to the soft tissue in our body. The collagen protein can be found on a subcellular level in the extracellular matrix. Additionally, it can be seen in bone cells and skin cells. Lastly, collagen can only be found in animals. So in type 1 collagen, the protein gains a new function because there's a substitution of cytosine instead of glycine in the triple helix, which changes the overall function since the structure changes. So this is the model that I made. I have it on my iPad. I made it out of Play-Doh earlier, but it's two different models, and I had to get rid of the Play-Doh for friends, so I have photos of it. So in the normal collagen protein, you'll see the triple helix as stated before, and the triple helix is called by the glycine, which is added, and it basically strengthens the helix and makes it have that shape. And then you'll see other amino acids like proline, hydroproline, and then the glycine, which is added. Whereas in your mutated one, it starts out with the glycine being there, but then you'll see cytosine put in, which causes the line to go flat, and it changes the overall shape of the collagen protein. So it's no longer that trihelix shape that we see before. It flattens out to be like one singular line, and you can see other amino acids cause a mutation in it, like alanine, valine, and serine. It can block or be substituted in for that glycine and cause the line to flatten out like that. So now I'm going to dive a little deeper and talk about the amino acids. So the main functional group presented in the original amino acid side chain is the amino group and the carboxyl group. The original side chain of glycine is nonpolar, and then the original collagen side chain is neutral. The original collagen side chain, when compared to the 19 other amino acids, it's considered small. When you look at the mutated amino acid side chain, you'll see the amino group, carboxyl group as well, but you'll also see methyl groups and the isopropyl group. The new mutated amino acid side chain of cysteine is slightly polar, and then the new amino acid is still neutral, but when you compare it to the 19 standard amino acids, it's a lot larger. So in the original side chain of collagen, we see the presence of glycine, which is smaller and nonpolar. However, in the new chain on the mutated type 1 side chain, the cytosine, which is substituted for that glycine, is larger and nonpolar. And again, other amino acids, like I listed before, can also lead to mutations when put in. So the alanine and valine or serine can also cause mutations on the original trifolds. So you can go back to this shape when other stuff's added, but we see that when a larger amino acid is added, it leads to the mutation since the helix shape can't be maintained. So the impact of collagen type 1 is that it will prevent structural support in the tissue, and then the mutation can lead to cell disruption and of collagen fibers, and this can lead to brittle bone disease like osteogenesis imperfecta, which is where your bones are brittle and they're more fragile, which means they tend to break. And we can see issues with connective tissue in general, like the tissue surrounding your organs, like your heart. It may be very weak or not as strong. So some limitations to my model is I don't show the actual breakdown of the amino acids. As seen in some models, you'll see the specific amino acid breakdown. And then also I don't show the whole amino acid chain because it's a lot larger than this, but I condensed it down to make it smaller. So that's my project. Thank you.